everyone, and welcome to another session of Breakfast at UMHealth. So thank you for joining us today, early and bright. So I would like to introduce our first speaker. So we have Dr. Liu Beifen. So she is an associate professor at the Department of Molecular Medicine, Faculty of Medicine, and she's also a research fellow at the Nanotechnology and Catalysis Research Center, NanoCat. Dr. Liu earned her PhD from the London Centre for Nanotechnology, Imperial College, London, and her research focuses on developing portable, rapid, and sensitive point-of-care devices for biomedical applications. Her team's biosensor innovation has received multiple prestigious awards, and she has published extensively with over 90 scientific articles and book chapters to her name. And she has also contributed internationally, serving as reviewer for journals and a technical committee member in ISO and OECD standardization efforts related to nanomaterials. Okay, so today she will be speaking on nano-flexible electrochemical biosensors for rapid point-of-care diagnostics. So while the presentation that we're about to watch is pre-recorded, as Dr. Liu is currently traveling, she's in an airport now, so she will be joining us live later for the Q&A session. So without further ado, let's begin the talk.
So here shows the outline. We will start with a fabrication of flexible electrode, so how to develop the nano thin film electrode, follow with the introductions of electrochemical biosensor, how we can develop the biosensor for the foodborne pathogen detection and also early cancer detections. Lastly, we will discuss about the lateral flow immunoassay, which is a paper-based biosensor. So we will discuss about the component and also the constructions of the lateral flow strip. Yeah, so this slide shows the key element and working principle of the biosensor, especially for reductions, rapid sensitive intermolecular interaction detections. Yeah, so at the heart of any biosensor actually is the biomolecular interactions. Yeah, so these interactions are generally non-covalent binding, which is very important for achieving the low limit of detections. Example include the antigen-antibody pair, antigen aptamer, biotin avidin, protein and its binding protein. Yeah, so finding the right molecular pair actually is not easy, especially for the novel molecule or the virus particles. Yeah, so as you can see from these figures, here shows the element of a biosensor. Actually, biosensor can work where it can detect various kinds of sample, for example, the cells, human sample including blood, urine and saliva, food samples, and even environmental samples. Yeah, so here shows the transducer. Transducer include the bioreceptors such as the nucleic acid, cells, antibody, aptamer, and enzyme, yeah, and also electrical interfaces. So these bioreceptors will combine with electrical interfaces such as FET devices, nanowire array electrode. Yeah, so the transducer output will be amplified by the electronic system, so it can be processed, displayed as a measurable data. So, but the bottleneck for biosensing probe development, the first bottleneck is a molecular interacting pair. As mentioned just now, for the novel target, finding a suitable molecular partner with high specificity and affinity actually is always the slowest step. Yeah, and then the second bottleneck is for the probe fabrications. Yeah, so the BIOS conjugation attaching the bioreceptors to the transducer surface while keeping it functional and stable is a rate determining process. It's a slow process. Yeah, so for electrochemical biosensor, the electrode surface preparations and functionalization are important to determine the sensitivity, the reproducibility, and long-term stability of the biosensor. So here shows the 3 working electrode for the electrochemical biosensor. And then if you are familiar with the biosensor work, you will see some of the lab are using the This is a glassy carbon electrode, yeah, and some of the company, they would like to commercialize it, so they will use a single printed electrode, yeah. So the key feature of this electrode need to be like multi-product. It can be multiplex, yeah, can detect multiple sample at the same time, yeah. Short detection time, sorry, there's a typo here, short detection time, yeah, stable, high sensitivity, low detection limit, and it's low cost, simple to use, and you can bring it everywhere with you. It's portable. So in our lab, we have a collaboration with NYCU to develop the palladium nano thin film fabrications and to use it for different kinds of the biomedical applications. You can see here, this is the example of a palladium film, and here shows the top view of the palladium nano thin film on the PET substrate. Here shows the cross sections, and you can see the palladium nano thin film is on the PET substrate. Yeah. Here shows the SRD spectrum of a palladium nano thin film electrode, and this is AFM images of the boundary between the palladium nano thin film and the PET substrate. Yeah. So we also ran the electrochemical study like CV curve to study the electrochemical stability and how stable this electrode is. Yeah. So you can see here, this is the PET, this is the palladium nano thin film on the PET substrate. Yeah, we use it as a working electrode, so you can see you can put 4 samples and you can detect 4 samples using one. So then we try to study the CV curve, that the stability of this palladium nano thin film. You can see that the study shows that the palladium nano thin film exhibit a peak potential difference of 74 millivolt, which is similar to the standard gold electrode as shown here. So this study suggests that the developed palladium nano thin film exhibit the electrochemical properties that are comparable to those of a gold electrode. So this study shows, we also continue using this palladium nano thin film to detect the DNA-based anti-cancer compound such as doxorubicin, F4N, and also the Taxol. This interaction is where we are using the solution, the ferrocyanide, and then we only use 1 microliter of anti-cancer compound, and we can get the reading within 11 minutes, and the sensitivity can go down to 0.1 micromolar as detected by the EIS spectrum. You can see here. So we believe that this highly reactive surface can also of this working electrode can also be used in other kind of the biomedical applications. So this is the images that we carry out the sputtering test in the NYCU, and then these are students, and then after the sputtering process, we further continue using this palladium nano thin film for the electrochemical study. And we also use this, and Prof Q is here, and we closely collaboration with our collaborator, Prof Chang here. So, and these are the paper published in the Biosensor Bioelectronic. Yeah, it's on the rapid screening of SARS-CoV-2 inhibitor. As we know that SARS-CoV-2 enter the cell through the binding of its spike protein, the S protein, yeah, to the cell surface expressing angiotensin converting enzyme 2 ACE2. So the inhibitions of the S protein to the ACE2 binding may slow down the SARS-CoV-2 cell entry and reduce the progressions of the COVID-19. So in this study, we use an electrochemical impedance spectroscopy, the EIS, based biosensing platform consisting of a recombining ACE2 coated palladium nano thin film and a core sensing element. This fabricated to screen the potential inhibitor against S protein ACE2 binding. So besides that, we also working on to fabricate the nano-flexible electrochemical biosensor for foodborne pathogen detections. Yeah. So recently we just published a paper on the ACS journals. Yeah. Then in this review, we look at how nanotechnology is used to detect the foodborne pathogens foodborne bacterial pathogens. It covers different type of the nanomaterials, the metallic, the non-metallic, and also the inorganic. So this highlight the unique properties of the DNA and non-DNA based materials for biosensing applications. You also include how DNA nanotechnology improve detection sensitivity and accuracy. So why we choose to study the foodborne pathogen detections? Because after interview with a food safety officer from the MOH, they informed us that they generally send like 1,000 samples from every round of inspections to the public health lab and also their in-house lab. Yeah. So to analyze the bacterial contamination in food, this lab normally will adopt the molecular method, which include the DNA extractions and follow with the pulse field gel electrophoresis or multiplex PCR or qPCR. So imagine if you send 1,000 samples in the same time, normally they will receive the report after 30 days. Yeah, so this is very time consuming and it's costly, and it may delay the response, lead to outbreak. So in this study, we try to develop the singleplex where you can detect one sample at the same time, or multiplex reader that you can detect several samples at the same time. So the concept is you need a strip, yeah, follow with an electronic reader, can be singleplex or the multiplex, and this reader can connect to the handphone through the Bluetooth. Yeah. Then the conductive sensor strip, as you can see here, actually normally will contain a recognizer molecules, and in our case here is aptamers. So this aptamer will bind to the outer membrane protein of the bacterium. So for the working electrode, we use the graphene. So maybe some of you will ask why graphene? Because graphene has a large surface to volume ratio, excellent electrical conductivity, yeah, high charge mobility at room temperature, and it gave high electron transfer rate. Yeah. So in our research team, actually we develop different kind of graphene-based nanocomposite. Can be RGO AP dye, RGO titanium nanoparticles, and RGO naphthalene and graphene-based conducting polymer composite. Yeah. So as we know that GO, even though it's hydrophilic, but it's very difficult, and it tends to aggregate due to the van der Waals force and by stacking interaction between the adjacent sheet. So if we use a GO, it's very difficult for us to disperse in the solutions. Yeah, so if we disperse it, like for example, if we make it into the nanocomposite, first it can enhance the sensitivity. Yeah, the second is it can help the GO to disperse well in the water. Yeah, so these are the development. So here shows the electrochemical biosensor. In this study, we use RGO, then we electrodeposit the titanium nanoparticles onto the working electrode, then we further conjugate with the aptamers. Yeah, then this aptamer is specifically target onto the salmonella in this case, and you can use different aptamer that specifically target onto the bacteria that you are interested in. So here shows the electrochemical analysis, and this is the CV curve, and this is the EIS. So the CV measures the current density, and EIS measure the resistivity. And you can see here the bare GCE, yeah, have a very high resistivity. Yeah, but then when we make RGO and RGO, even the RGO titanium nanocomposite, yeah, you can see the charge transfer resistance decrease significantly due to the effective electron transfer. So when we attach the aptamers, it further increase the charge transfer resistance due to the hindrance character. And then in the presence of the bacterial cells, the diameter of the EIS semicircles, you can see that increase further as compared to the one with the aptamers due to the charge transfer resistance. Yeah, so this EIS result is comparable with the CV. Yeah, so then we further continue our analysis on the sensitivity test. Here shows electrochemical detections of the bacterial cells using the fabricated electrode, and then with different concentrations of the bacterials from 10.1 to 10 CFU per mil in the Zubaidah solutions. Yeah, and then we try to plot the graph, the current density versus the different concentration of bacteria, and you can see the linear relationship between the density, the current density, and the cell concentrations here, with R squared 0.98 linearity for that. Then you can see from this graph that the detection limit can go down to 10 CFU per mil. Yeah, and then this is here shows that this is this sensor is developed for the salmonella, and this is the non-salmonella bacterial species, and those are the salmonella species. The Salmonella typhimura, Enteritis, Salmonella typhi. You can see that the biosensor, developed biosensor, have high specificity for the salmonella bacterials. Yeah, due to the specific aptamer that used, yeah, This slide shows the multiplex detection of bacterial in cell culture suspensions. Yeah, so we can detect multiple bacteria at the same times. So this shows a strip with 4 working electrodes. That means you can detect 4 bacterial in the same time. Yeah, so here shows a different model of the strips. Yeah, for the preparation of bacterial culture for lab testing, we will first use pure cultures, followed by full samples, which has been spiked with a bacterial strand. After testing this sample with a developed biosensor, then we will further verify the result using the gold standard method, including the bacterial plating and qPCR method. This slide shows the DPV analysis of different aptamers specific to different bacterial species. In this case, it's a Bacillus, SRs, E. coli, and Salmonella. Yeah, so the black graph is the aptamer only. Once bind with bacteria, the current density decrease. Yeah, so that's how we can set the threshold to differentiate the sample with and without bacterials. Then we try to think why we have to in order to decrease the cost, yeah, the cost of the fabrications, why not we develop the multiplex aptamers using the select method. So you can see that we try to develop aptamer for multiplex target, yeah, for cost-saving purpose using a process called the systematic evolutions of ligand by exponential arrangement select method. Yeah, so to generate a DNA aptamer, first of all we have a single-stranded DNA library with random sequence. So this library will incubate with a target. Upon incubations, there are certain sequence that will be able to bind to the target. After incubations, then a washing step is required to wash off the outbound sequence that cannot bind to the target molecules. After washings, the bound sequence will elute out and amplified using the PCR. So the select cycle will be carried out for a few cycles until the aptamer with high affinity with desired binding affinity are generated. Here shows we have found the patterns on the aptamer sequence for different types of the bacterials and compare their KD value with a transcated aptamer, the shorter aptamers, to evaluate to assess the sensitivity in biosensors. Here slide shows that we have sent our developed biosensor NanoSense to an ISO testing lab, and their staff help us to compare our biosensor result with the plating and also the PCR results. So one of the essential aspects of our evaluations involve comparing the performance of the NanoSense with an accredited method practiced by the Ministry of Health endorsed lab. So the full sample were tested using the accredited method used by the third party and then compare with our developed biosensor. And this shows our students managed to work together with the accredited lab. So our students will run the developed biosensor and compare result with the bacterial plating and qPCR result that was done by the staff in the company. Our developed biosensor NanoSense was tested with real food sample, and the result were verified using blood count and PCR method. While pure culture tests show 100% accuracy, but the performance with real food sample were affected by oil and other impurities. That's why accuracy here is lower, 80%. At the moment, we are trying with different sample pre-treatment method to improve the sensitivity of our developed biosensor. Yeah, besides that, we also work on the lateral flow immunoassay. Yeah, so this lateral flow immunoassay actually is the paper-based biosensor. Yeah, it's paper-based biosensor that we normally use, like we use it during the COVID-19. I think more of you know about this. Yeah, so this lateral flow immunoassay is usually we develop early cancer detections. Yeah, these are our students' key. Yeah, this shows a single plex direct lateral flow device. The sample actually is placed on the sample pad here, so it will flow through through the capillary actions. If the target is available, target analyte in the samples is available, so the detector molecules which is conjugated with a label, normally it's a 14 nanometer gold nanoparticles, they will bind to the target, and then it will flow through the test line, the test pad here. So the test line, actually you can see that there's a test line, also the control line. So the test line, the test line is modified with capture molecules, yeah, to capture the target analyte, while the control line is modified with the control molecules here. So this control molecule is served as an indicator to validate the test results, and will bind to the free detector molecule. Then the excess detector molecule will absorb by the adsorbent pad. So for the positive sample, you will see both line, yeah, on the test line, also the control line. Whereas for a negative sample, no target analyte here, so we only can see the control line, yeah. Then we also play around with different type of nanozyme. So nanozyme are the nanomaterials that act like an enzyme that turn the colorless substrate into a color product. Yeah, so that we can use a different nanozyme and see how it can enhance the sensitivity of the paper-based biosensor. In this presentation, I have summarized different kinds of biosensor, including electrochemical biosensor, such as a glucose sensor, and paper-based biosensor, which many of us use it during the MCO. So these biosensors have wide-ranging application in the biomedical field. Before I conclude my presentation, I would like to acknowledge my research team and students, collaborator, as well as all the funding agencies that have supported this work. Thank you very much for your attention. I'll be happy to take any questions. Thank you.
All right, thank you so much, Dr. Leo, for the presentation. So I think Dr. Leo is here to take any question from the floor. I think there's a question in the Q&A from Prof. Sarah Ahmad. She asks, from my limited understanding, it sounds like great work, saving time and money. Was this technique patented? Not sure which technique that she refers to. Maybe Prof. Sarah
Thank you,
is
Dr.
here.
Ai.
Hi, Dr. Leo.
Hi. Thank you for sharing the session. Yeah, maybe I can answer in general. Yeah, and actually this technology has been patented. For example, for the nanomaterials that we develop, the aptamer where we shorten the sequence, all these have been patented. Yeah, under UM.
So it's the aptamer that has been, the sequence has been patented. Is it? I see.
The aptamer for these three bacterials, and also how we develop this biosensor from the nanomaterials, and the detections actually we patent under UM. Yeah, we have patented technology.
Okay,
I hope I
great,
answered the
great
questions.
work.
Thank you, Dr.
I have another question too. I think you do a lot of different aspects of biosensors, right? Like from foodborne to cancer detection to the COVID-19 detection. It's a lot of like real world application, and that's making a lot of impact. I'm just wondering, right? What has been the challenges for you and your team to bring your research from the lab, from the bench to, application to commercialize it?
Yeah. I think that's a very important questions,
Mm.
Dr. Ai. Actually, when we run the testing in the lab, everything's perfect. And including the cancer, like for example, the cancer biomarker that we want in the lab is so perfect. But when in the real case, when we go to the clinical department, we test for the sensor, there are some specificity issues that we need to solve it. Like for example, for the foodborne pathogen, in the pure culture is perfect. But when we go to the field test, the sample may contain the contamination, the oily sample. All these we have to retreat. For example, we need to introduce a pretreatment to enhance the sensitivity of the sensor. So all these have to be taken into the consideration when develop a sensor. Yeah. But that's a very good question. Thank you.
Right. I think there's another question from the chat. Prof. David, amazing work. How many times can you use a single sensor?
Yeah. Actually, we are using the SPE, which is single printer electrode. So for this one, actually is one use, but the reader itself is multiple use. Yeah. The strip that insert into if you refer to my slide, the strip, that one is only one use. Yeah, but the cost is quite cheap lah. Yeah, it's not expensive.
And I think I have another questions to ask you.
Yeah.
Because I was looking at the slide just now, you work on different types of sample. I mean, different types of samples. Yeah. Have your team work on fecal sample?
Uh,
That's quite a difficult sample to work with, I think.
Currently, we're working on the bacterial samples. Yeah, some viruses and also the cancer biomarkers. Yeah, for team. That's how for this, like for a finger sample, maybe we can discuss in more detail. Yeah, as long as we can develop a bioreceptor, antibodies or aptamer for that, then you let us know what is the biomarker for that. That's how we can develop. Yeah, we can develop for this. No problem for that.
Yeah. Okay. Okay. Thank you so much for the presentation.
Thank you, Dodai.
Any questions from the audience? If there are no more questions, thank you so much, Dr. Leo, and I will pass the session to Dr. Vinish to chair the next session. Thank you.